Hello, everyone, and welcome to the Beggs Lab Nemaline Myopathy Research webinar. This webinar was funded through the Patient-Centered Outcomes Research Institute project in partnership with the Foundation Building Strength. Ellen, can you pop to the next slide, please?
Yep, sorry, working on it. For some reason, my keyboard is not responding. All right, cancel. Sorry, folks.
No problem.
There we are.
I just want to mention that this program is being recorded and it's going to be posted onto the internet, which is great for the people who couldn't make or attend the event at this time, but also for people that can refer back to it as a resource. Just the reason I want to mention it is because if you ask questions that are especially personal questions that might be related to your health or the health of a loved one, that this is not a private venue, and I wanted to make sure everybody knew that in advance. Thank you. Okay, so we could go to the next slide.
Sorry, everybody. Unfortunately, when GoToMeeting is active, I can't seem to advance my slides. It's a new problem. There we go.
Yeah, that's perfect. Great. Thanks, Ellen. I just wanted to mention that the Nemaline Myopathy Family Gathering is taking place this summer in New York City, June 24th and 25th, and I wanted to invite all of you to attend. The link is here to register, but I know this is kind of a wacky link to write down, so just rest assured that you could find this also on the Facebook page, pages for the Nemaline Myopathy families, and also it will be soon to be posted on the Foundation Building Strength website, and there will be emails going out, so you can refer to this link to register. If you have any questions, you can also email me, and my email was on the webinar announcement. So this is Sarah Foy. So with that, I'd like to go ahead and thank and introduce Alan Beggs and Casey Giannetti. Alan, should I go? Can introduce you guys, or did you want to introduce yourself?
Yep. Please go right ahead, Sarah.
Okay, so Alan Beggs he's the director of the Manson Center for Orphan Diseases Research at the Children's Hospital Boston, or Boston Children's Hospital. And he has directed an independent research laboratory in the genetics division at Boston Children's Hospital since 1992. And the aim of his research is to understand essentially muscles and muscle diseases. And he's very well respected. And as a affected family, I've been known Alan for many years and really appreciate his work. And so I'm really looking forward to listening to him today. And along with Alan is going to be Casey Giannetti. She's a genetic counselor and project manager at Boston Children's Hospital, specializing in rare disease research, genomic sequencing, and gene discovery. So thank you guys so much for taking time out of your weekend to be with us, and I'm going to go ahead and turn it over to Alan Beggs. Thank you.
Thank you, Sarah. It's great to hear your voice, and I'm really thrilled to see so many familiar names logged into the webinar today. Hi to everybody who I know personally. It's great to see you. And to the folks who are new, I look forward to chatting and interacting with you now, and hopefully meeting you sometime in the near future. So what we thought we would do today is, some of you have seen some of this before, but for others it's maybe a little bit new. I want to start with kind of talking a little bit about what is Nemaline myopathy. Nemaline myopathy is really a number of different things, but they're all highly related, have common links. And I'll tell you a little bit about that. Talk a little bit about the work we've done for the past 10 years or so, finding the genes that cause Nemaline myopathy and what some of those genes are. And then Casey Giannetti is going to talk a little bit about our enrollment in our research project. Many of you are already enrolled in that and have contributed tremendously over the years. Without that participation, it would be impossible to do the research that we've been doing. And then finally end with a description of some work we're doing using zebrafish as a model for Nemaline myopathy. And I'll try to explain why this is so important, in particular for our development of therapies they've become really critical in our search for drugs to treat Nemaline myopathy. So to start, what is Nemaline myopathy? Well, this is a picture actually from the patient conference we held in 2009 in New York. It's actually the same conference series that Sarah was talking about a few minutes ago. And you can see everybody in this picture here has Nemaline myopathy. You can see there's a lot of variation. Some of the children are unable to sit up on their own or have breathing assistance, and in fact some of the very sickest kids weren't even able to travel to this. And yet some of the folks standing in the back are obviously still able to walk. Many of them grow up to have children and grandchildren, and so that clinically speaking there's a tremendous variation in the severity of the condition depending on how much weakness there is. It's what we call a congenital myopathy. It simply just means that you're born with it. Myo is muscle and pathy is pathology or disease. So it means a muscle disease that you're born with. It's relatively rare, as many genetic conditions are. It has an incidence we estimate of about 1 in 50,000 births. And it's characterized by what in the literature has been said to be non or slowly progressive muscle weakness and hypotonia. Hypotonia is loose muscles or floppy muscles. I should say that it does progress somewhat over time, as I think many of you probably have experienced, but it's considered less progressive compared to things like muscular dystrophy, where, for example, a boy with Duchenne dystrophy may be able to run and walk at the age of 4 to 5, but then often needs a wheelchair by 11 or 12. As I mentioned, the severity can be quite variable, ranging from mild weakness to adulthood, in childhood, sometimes ventilator and wheelchair dependence, and then severe arthrogryposis, which means contractures, so very severe weakness at birth. This just shows what some of the muscle looks like under the microscope. On the left are light micrographs of 2 different muscle biopsies from individuals with Nemaline myopathy. And this particular stain normally would stain the muscle fibers. That's what you see here, these polygonal structures. They're really long tubes coming out of the picture at you. And normally it stains it just a uniform pale blue. And you'll see in both of these some darker, granular, really dark structures. These are the so-called Nemaline rods, which give the condition its name. If you look on the right-hand side, and these are pictures of electron microscope pictures at a much higher magnification. In the lower right here, you see this repeating structure that characterizes skeletal muscle, and then these dark rod-like structures of the Nemaline rods. So those are the same things that are staining dark blue over on the left. And you can see that where the Nemaline rods are, they're disrupting the normal zebra stripe pattern of skeletal muscle, and therefore reducing that muscle's ability to contract properly. The 2 pictures on the bottom here come from an individual who had somewhat milder weakness. The 2 pictures on the top come from an individual who had more severe weakness. And you can see in the top right here that the nice zebra stripe pattern is really very highly disrupted in some of these muscle fibers. So over the years, we've been studying the genetic basis for this, and we've discovered a variety of genes that can be mutated in individuals with Nemaline myopathy. So an abnormality in any one of these genes can cause the same picture under the microscope in the same clinical picture. And it turns out there's a nice unifying feature here that these genes all encode components of the so-called thin filament, which is shown in detail at the bottom. If you look in the middle here, we have a diagram of the sarcomere, which is the contractile unit, or the basic repeating unit of skeletal muscles that contracts to cause a whole muscle to contract. And you see these dark gray structures, the so-called actin thin filaments, and then in the middle these red structures of the so-called myosin thick filaments. And these myosin heads reach out, grab the actin, and pull it inward in both directions. So this side slides in, and the left side here slides in, and together that shortens the distance between these 2 vertical structures, the so-called Z-bands. And muscle is composed of a large series of these sarcomeres in a row, and so when each one contracts a little bit, the long fiber actually contracts measurably, and that's what allows our muscles to move. What we've discovered in Nemaline myopathy over the years is that virtually every gene that's capable of causing Nemaline myopathy encodes either a protein that makes up part of the actin thin filament, shown at the bottom, or, as I'll show you in a few minutes, sometimes encodes proteins that we now are learning control the structure of the thin filament. So they may not be part of it, but they actually affect the maintenance of that structure. So to summarize then, Nemaline myopathy, I often tell medical students, and this can be somewhat confusing to them, but heterogeneity rules the day. It actually makes our life a little bit harder because we need to understand all the different variations of it in order to develop therapies. But clinically speaking, there's basically a continuum ranging from children who are incredibly weak at birth and may not even survive the first few years of life, all the way up to individuals who are able to walk, who need only minimal breathing assistance, if any, and who, as I mentioned earlier, can grow up and have grandchildren and live a relatively more normal life. As I showed you under the microscope, there's heterogeneity or variation in the pathological findings, how the muscle looks. And then, as I just mentioned, there's genetic heterogeneity. Basically, there are different genes, and a mutation in any one of them leads to the same defect in the thin filament, and therefore leads to the same medical condition, Nemaline myopathy. And I didn't touch on that, but there are multiple inheritance patterns. So in many cases, the mutation that causes Nemaline myopathy may be a new or de novo change. De novo changes in our DNA are happening every time a baby is conceived in all of us, but every once in a while it might alter a gene that causes disease, in this case one of the Nemaline myopathy genes. So in that situation there may be no family history, no relatives who have any condition related to Nemaline myopathy. In other cases it can be a milder mutation that's what we call autosomal dominant, where you have one abnormal copy that's passed down from parent to child. And then there can also be what's known as autosomal recessive inheritance, where you need to inherit two abnormal copies of a gene, one from each parent, and the parents are normal, having only one abnormal copy. And so in this case, families at risk where each parent may be a carrier, they may have roughly 25% of their children will be expected to inherit the disease. So I want to talk for a few minutes about some of the genes that cause Nemaline myopathy and really how we find them. And then Casey will tell you a little bit about how that's all been made possible. So this has really come about through our congenital myopathy study that I think many of you have participated in. We started this way back in 1992. As Casey will explain, we identify and enroll patients and their families from around the world. So this is not limited just to Boston Children's Hospital or just to North America. But in fact, it's been a collaborative effort. We have individuals that enroll directly in my lab from almost every continent, nobody from Antarctica yet. And we also collaborate with individuals in other parts of the world. So some of you probably know of Nigel Lang in Perth, Australia, or Karina Wahlgren-Pedersen in Helsinki and other groups. And in sharing this it really allows us to magnify the value of each patient's research participation. This is a somewhat older slide. We now have probably over 3,000 individuals enrolled. Of these, 700 or 800 have a congenital myopathy, and I think we're approaching roughly 350 who have Nemaline myopathy specifically, and the other families have related conditions, such as what we've worked with Sarah Foy and her family. And as Casey will explain, we collect clinical records, DNA, and muscle specimens. So doing this has allowed us then to search the DNA for some of the genetic causes. So I want to just quickly go through a few of the different ways that we've done this over the years and how this has evolved. So the first way is a so-called candidate gene approach. What that means is that, as you're probably aware, our genome is very complex. We have roughly 3 billion base pairs. or bases, individual units of information in our DNA. And encoded within those 3 billion bases are roughly around 20,000 different genes. So when we're faced with an individual with a condition like Nemaline myopathy and we don't know what to look for, we can't, until recently, it hasn't been possible to search through most of these genes, and so we had to be selective. But in this case, knowing, for example, that some of the genes that encode proteins of the thin filament here, knowing that some of those cause Nemaline myopathy, we could hypothesize that some of the other genes that we hadn't looked at previously might also do the same. So for example, these purple balls here encode a protein called cofilin. And when we studied this family, whose pedigree is shown in panel A here, you can see that there were 2 girls, circles that denote girls. These 2 circles are filled in. They both had Nemaline myopathy. They were the children of 2 cousins who had married each other. They were a family from Bahrain. And as it turned out, we didn't know the cause of their Nemaline myopathy, but we knew at that time that, for example, mutations in the actin gene, which are these gray balls, or mutations in the nebulin gene, which is this green snake-like structure here, we knew they caused Nemaline myopathy. So Dr. Pankaj Agarwal, working in my lab at the time, hypothesized that maybe cofilin, these purple structures, might also be a cause of Nemaline myopathy, knowing that these other closely associated proteins could cause it in the first place. And in fact, that was the case. The lower right-hand corner here is just showing the DNA tracing, and you can see the normal sequence is a black or peak denoting the letter G, which is one of our DNA bases here. That's in a control individual. And as it turned out, one of the 2 sisters, and in fact both of them, had only a green peak here, denoting a change to the letter A from G to A. That was the mutation. You can see that the father here had 1 copy of each of these. And long story short, we predicted that this affected the protein by changing an amino acid at a critical structure within there, and went on to prove that this was the cause of Nemaline myopathy in that individual. I mentioned the actin gene, and I'm going to talk about that in a little bit later in the talk, but this is a schematic of the actin gene, and it shows each of the square rectangles here is part of the protein coding portion of the gene, so the business part of the gene. And each of the arrows underneath indicates the location of a different mutation. So one of the things we've learned is that some patients may have a Nemaline myopathy caused by a mutation in cofilin, others by a mutation in nebulin, and yet others caused by a mutation in actin. Excuse me. And but not only is there variation in which gene is mutated, but the location of the mutation can change. And so all these differences are partly what contributes to the difference from one individual to the other in terms of severity, patterns of weakness, and so on. Now, another gene you've probably heard quite a bit about, I know some of you are familiar because this is your gene, is the nebulin gene. Just to compare nebulin to actin, this diagram here shows the entire actin gene. And if we count up the coding region, it's 3,561 bases long. For comparison, the nebulin gene is over 250,000 bases long, so it's extremely large. Another comparison, the actin gene has 1, 2, 3, 4, 5, 6, 7 exons, or 7 protein coding regions. So the business end of actin then is broken into 7 pieces. In contrast, the nebulin gene has over 180 exons. So what this means is that searching the nebulin gene for mutations is very much harder than it has been for actin, and it's one reason why there are individuals who only recently have had their nebulin mutations identified, and there are still others who we're searching at this point. So one of the revolutionary developments that's occurred in the past 5 years or so in human genetics is the development of what's called next generation DNA sequencing. So sequencing the actin gene, when we found those mutations, required a targeted experiment that looked just at one tiny little piece of the gene at a time. And to sequence the entire gene took a person maybe a week to do, and they were looking only at a single gene. This rather complicated diagram illustrates a very different way of sequencing our DNA. Essentially, all of the genomic DNA, or all of our DNA, is broken up into tiny little pieces, and then those pieces are independently sequenced in parallel. So there are companies that have developed these so-called next-generation sequencing machines. They sequence hundreds of thousands of different little pieces of DNA simultaneously, and then a computer lines them all up with each other and assembles them. And the result is that instead of sequencing one gene in a week, you can sequence all 20,000 genes in a week. And this has really revolutionized our approach to finding new genes. So one of these genes, in fact, is a gene that's called KLHL41. I apologize, the slide seems to have a little problem here with the text, but this is a gene that was discovered by Donna Gupta, who is now an assistant professor with her own independent laboratory at the Brigham and Women's Hospital. But while she was working in my lab, in a collaboration with Nigel Lang, Karina Waldron-Pedersen, and a large international group of other investigators, sequenced using next-generation sequencing the DNA from a group of individuals with Nemaline myopathy, and identified mutations in this new protein KLHL41. So the slide is entitled KLHL41, a new paradigm of disease. Let me just spend a minute explaining why we think that's true. Most of the genes that cause Nemaline myopathy are shown on the right here are labeled. The Nemaline gene, which is this long green snake, the actin gene I've mentioned, these gray balls. This troponin complex is actually 3 different proteins, and we know that troponin T mutations can cause Nemaline myopathy. Cofilin mutations can cause it. A protein called LMOD3, which helps cap the end of these thin filaments, can cause it. But then about 5 years ago, another protein called KBTBD13 was identified. And Nigel Lang's group and our group together simultaneously identified mutations in KLHL40, and as I mentioned a second ago, KLHL41. Well, these proteins are actually not known components of the thin filament. So the question was, what link did they have to this common structure that we thought was responsible for Nemaline myopathy? And in work just in the last couple of years from Vandana and from some other groups around the country, we started to understand that the functions of these K proteins, that K stands for a word kelch, which is a common structural motif these proteins have, that they are involved in the stability and the turnover of the particular proteins found here. So for example, KLHL41, we now know, actually binds and stabilizes Nebulin while it's being synthesized. And may play a role also in helping to turn over Nebulin as it gets damaged with age. So the ability to rapidly sequence our genes has allowed us to identify all these various things. After Casey talks, I'm going to talk a little bit about zebrafish, and the information we have about what these various proteins do is actually allowing us to develop therapeutic approaches that we hope are going to lead to treatment in the future. So I'm going to let Casey talk for a few minutes now, and she's going to kind of discuss a little bit how we do this, and then I'll come back and tell you about the zebrafish work we've been doing. Casey?
Hi, everyone. Nice to meet you for those who I haven't met previously, but I think quite a few of you I've had the pleasure of speaking to and enrolling in our lab. And some of you may have worked with my predecessor, either Lindsay Swanson or Elizabeth Duchenne. But for those of you who are not already involved in the BEGS lab, I'm going to give you some information on that as well. So Alan, do you want to go ahead and switch to the next slide?
Yeah, I'm sorry. I just muted myself, and every time I click on the GoToMeeting control panel, then I lose, there we go, the ability to control the slides.
All right. So Alan was talking a little bit about how we do some of our gene discovery, and that's one of the aspects of what we do in terms of the clinical research. And a little bit later in this talk, he's going to go ahead and talk about some of our lab work, which happens down the hall from where I sit in our kind of department at Boston Children's. So in terms of the clinical research, everything starts with our patient enrollment. So for those of you who are already enrolled, you're familiar with this, but for those of you who aren't, we typically start with what we call our proband. So that's our affected individual or patient, and we enroll them in our lab as well as their family members. And I'll talk a little bit more on the next slide about the actual enrollment process. But once someone is consented and enrolled into the lab, we do a few different kind of action items in terms of what we need to collect from that family. So first would be a sample collection, and that can be DNA from either blood or saliva, and then we can also try to get a tissue sample, and that would be from either a previous muscle biopsy that was done. Many pathology labs have the practice of storing frozen tissue or slides that were used to prepare a muscle biopsy pathology analysis, so we can oftentimes request some of those materials to store in our lab. And then we also try to collect a lot of clinical data. That can be actual medical questionnaires and family history questionnaires that we do on the phone or in person, as well as medical records and pathology reports that can help us get a really good picture of someone's kind of their clinical history. And many of the patients and families that we enroll, we never have the pleasure of meeting because, as Alan mentioned, they're from all over the world. So we rely very heavily on this type of clinical data. Now, once we have all of this in our office, some of the projects that we work on can really range in terms of whether we're using the samples that we've collected or some of that clinical data. So in some cases, we're actually doing that genetic testing or gene discovery, as Alan was just speaking about. So this would be looking at both the patient as well as their affected or unaffected family members to try to identify genetic changes in any of those genes that were previously mentioned, as well as possibly new genes or genes that may have not been previously associated with Nemaline myopathy. We can also do what we call functional analyses. And that's basically helping us to prove whether or not specific genetic changes are actually impacting how the muscle is functioning. So sometimes we are doing that in cell lines, which can be created from the blood or other tissue samples, like parts of the muscle that can be turned into a cell line. And so we do that in a dish, looking at different expression of proteins, and trying to give us a little bit more information regarding whether certain genetic changes are actually causing a problem or actually leading to those Nemaline rods. This can be really helpful in cases where we have found a genetic change that we're just not certain of, or something that we don't have enough evidence to say that it's actually leading to disease. So that's what we do with some of our samples. But another big area of our lab, we're using the actual clinical data, and this is in different things like natural history studies and cohort comparisons. So something we've been working on recently are looking at the whole Nemaline group as a whole, and then kind of breaking it down into individuals who have different genetic changes and trying to see if certain gene changes are related to more severe or more mild phenotypes, and if we're able to predict any of that by just looking at the genetic changes themselves. Natural history studies are extremely important part of the way we are able to develop different therapies. And so it's really important to have a very good understanding of a condition itself before we can even start determining whether particular therapies are working or beneficial or not. And so we can do a lot of our chart review and data collection to help us get a really nice history of what Nemaline looks like. And Alan, if you want to flip to the next slide. So enrollment itself, for those of you who are not previously enrolled, we look to enroll individuals who have a suspected or confirmed congenital myopathy. So this could be individuals who've had a muscle biopsy that shows an abnormal pathology, or it could be individuals who may not even have had a muscle biopsy yet, but other types of muscle disease have been ruled out, and there's a strong suspicion for a congenital myopathy. And we can also enroll individuals who have already had genetic testing as well that tell us that they have a Nemaline myopathy. And the enrollment can be done here at Boston Children's. We enroll many individuals from our neuromuscular clinic, along with some of our neurologists who see the patients clinically. But we can also enroll individuals from all over the country and all over the world. So we do a lot of speaking via phone or Skype to complete our consent process. This is basically going over paperwork that talks about the benefits and risks of research and important things like data security and really just some background on kind of what we are able to do with this type of research. And then we can also even collect things like samples, medical records, and tissue remotely by working with pathology departments and shipping things back and forth. Along with that consent, we get medical record releases. We have a verbal consent phone call with all individuals over the age of 18. And even with the kids, once they're 7 years old, they have kind of an abbreviated phone call with me or one of my colleagues to just make sure that they understand what research is and make sure that they're okay with sending in a blood sample or a spit sample or having someone look at their medical records. And then as we mentioned briefly, we try to collect some family history to see if there are extended relatives that might be important for us to have enrolled in the study, and then also some different medical history questionnaires. Over time, we've started to make some new questionnaires, and so families may know that we oftentimes will recontact to try to collect some new information or to get updates because perhaps a family was enrolled 10 to 15 years ago and we want to see how things are looking now. So having that kind of longitudinal data analysis is extremely helpful. So for those of you who are not enrolled and who are interested in being enrolled, my email address and my phone number are there, and you can always reach out to Sarah as well, and she has all that information. And same with individuals who are already enrolled. If it's been a while since you've been in contact with us, or if you have some new clinical data or perhaps new testing results that you'd like to share with us, we always love to get updates. And so you can reach out via email or phone as well, because we certainly love to hear from you. And you can go right to the next slide, Alan. All right, so I just wanted to also touch back on the genetic testing for Nemaline. This is something that historically we've done a lot in our lab, but it's also something that's available a lot more clinically now as well. So I just wanted to touch on that a bit. First of all, when someone has a muscle biopsy that shows Nemaline myopathy, that obviously we would call a clinical diagnosis, but oftentimes it can be very helpful to have a molecular diagnosis or to know the genetic changes. That information can be helpful for family planning or recurrence risk. It can also guide clinical management in some cases, and probably more and more in the future, it's going to help to impact eligibility for future clinical trials and therapies as we start to look at therapies that are more specific for certain genetic changes. So if you are an individual or a family who has never had genetic testing, probably 5 or 10 years ago, this is something that was done much more commonly on the research side and not as much in the clinic, meaning not as much through a neurologist or a geneticist. But now that we have really discovered a pretty large panel of genes that are known to cause Nemaline myopathy, this is something that's being done much more frequently by a clinical neurologist or geneticist. In laboratories like University of Chicago, Prevention Genetics, GeneDx, there are many, many that we could list off, actually offer panels. So that's that next generation sequencing that Alan was mentioning. It's a very effective way for us to quickly and fairly inexpensively look at a handful of genes or even more than a handful of genes all at one time. So if that's something that you have not previously done or previously talked to your provider about and you're interested in getting a molecular diagnosis, I think that's a great place to start. I am always available as a genetic counselor to answer questions about that type of testing or even answer questions for providers who might not be as familiar with genetic testing. I am a lab or a research genetic counselor now, but I've trained in the clinic as well. So that's one thing that can be done through your clinical neurologist. But I also get a lot of questions about negative testing. So what happens if someone has one of those panels and there is either no result or the result is what we call a variant of uncertain significance, or just not a clear answer. So we do, at this point in time, have even larger options. Some of you may have heard of something called whole exome or whole genome sequencing. It's a technology that lets us look at all of the genes at one point in time. And that can help us to look at additional genes that we may not have been well associated with Nemaline in the past. This is something that is starting to be done more frequently in the clinic, but it's something that we also do pretty regularly on a research basis. It's a bit more expensive, so it's something that we're starting to try to utilize, especially for cases that have had previous testing of some of our known genes and are still negative. So in those cases, for many of the families who we have enrolled, we always try to recommend that you speak to your provider about getting some of the baseline testing done, and then we are able to try to take it a step further in many cases where there's negative findings. Certainly, we recommend families seek us out and enroll regardless of what testing you've had done. It just sometimes can be helpful to have some of that baseline done before we jump to some of these larger technologies to try to look for additional genetic changes that might be causing Nemaline myopathy in a family. Alan, I don't know if before you pick things back up, if we want to see if anyone has questions about the genetic testing or some of the genes involved before we go into some of the new research on therapies.
Yeah, I think that's a great idea.
Any questions for anyone, for either me or for Alan, about enrollment or some of the history of Nemaline or the genes involved? And I think Sarah's saying that if you, as we're continuing, if you have any questions that you want to type into the chat, feel free to do so. And also if there are things that come up later after this webinar, always feel free to reach out. So Alan, why don't you go ahead and take back over.
Okay, great. Well, thank you very much, Casey. So, yeah, as Casey said, now we're able to find many of the genes that cause this. We're actually still actively researching because there are maybe 20% of individuals who we test that we cannot find a genetic cause for their Nemaline myopathy. But shown here is a summary of the 10 really well-established genes that can cause Nemaline myopathy. And in the last year or two, there have been reports of one or two, at this point, sort of oddball genes that we're not sure how they fit in and not quite sure if the person has Nemaline myopathy or not. But these are the 10 well-established genes. And as you can see, Nebulin, NEB, is the most common cause. In our group, in our study, we've identified Nebulin mutations in about 40% of our individuals. The mutations of the actin gene are the next most common cause. Roughly about a third of individuals have those. And then these other genes, TPM2, TPM3, and so on, they all account for a few percent, so a very small percentage of all the cases. Well, this is obviously great. There's a lot of benefits that can come from knowing the basis for your Nemaline myopathy, for understanding the risk for recurrence. But really our long-term goal is to develop effective treatments so we can actually do something proactive. We've focused primarily initially on Nebulin and actin because these are clearly the two most common causes. Well, some of you may have heard about gene therapy. There's a trial that we hope is going to start soon for a condition called myotubular myopathy. We've certainly thought about gene therapy as a possibility for Nemaline myopathy, but there are differences between these conditions that make it problematic. So the first is that, for example, the Nebulin gene is extremely large. I showed you has over 180 exons. And so the forms of gene therapy that are possible today don't allow for the transfer of such a large gene. Unfortunately, Nebulin is an order of magnitude too large to work in that type of a system. The actin mutations, on the other hand, the actin gene, as I showed you, is relatively smaller. That could theoretically fit. However, you'll note that a lot of the variants or the mutations that cause actin Nemaline myopathy are what are called dominant. So in effect, those individuals have one abnormal copy of the gene and one normal copy of the gene. And the Nemaline myopathy is actually caused by the negative impact of that one abnormal copy. So in the case of actin myopathy, it's not a question of adding back something that's missing, but the problem is we really need to think of ways to take away or counteract the effects of something that is present and actively having a negative impact. So the approach that we have favored, and an approach that has really become very well established in recent years in genetics research, is to use zebrafish as models of developing therapy. A lot of people express quite a bit of surprise and uncertainty about why we would use zebrafish as a model, but I hope to explain to you some of the powers of this system. So first of all, zebrafish are relatively small, so we can grow large numbers of them. Their husbandry is relatively easy, so in fact, in comparison, for example, to mice, which may take several years to study, cost hundreds of thousands of dollars, and you may study 10 or 20 mice, we can actually have tens of thousands of zebrafish all growing together at the same time. They're a great model also because they have high spawning productivity. So these are zebrafish embryos at about a day and a half of development. And each clutch of eggs, you take 2 zebrafish, and when they spawn, they'll produce a clutch with about 100 eggs. Those eggs develop very rapidly and ex utero, so in other words, as an egg outside the body, which actually allows us to watch it. So you can see in the bottom here is a series of photographs of an egg developing, starting from a fertilized zygote, just that egg that's just been fertilized, through to hatching at about a day and a half. And then by 2 days, 48 hours after fertilization, you actually have a small free-swimming fry. And along the way, these embryos are clear, so you can watch them develop. And so if you, have, for example, have a defect in, for example, Nemaline myopathy gene, you can actually see the effects of that in real time. Finally, despite the fact that fish and humans appear outwardly so different, there are really a tremendous number of similarities in the way that our muscle is built and organized, and we have the same sets of genes that control the muscle. So for example, if you look at a zebrafish in cross-section, as you know, if anyone has had salmon or swordfish for dinner, most of that fish is muscle, and the muscle is all alongside the flanks of the body here. When we look at the various proteins, we find that the genes and the proteins are the same. So they're not identical, obviously, but we have, for example, the same dystrophin protein in zebrafish that when mutated causes Duchenne muscular dystrophy in people. And when we look at high power under the microscope at the sarcomeres in a zebrafish, they have the same structure. Zebrafish have an actin gene. They actually have 2 actin genes. They have a nebulin gene. They have a tropomyosin gene. So again, all the same genes that are present in humans. And this allows us to study potential treatments on a much larger scale and much more rapidly to identify, for example, drugs or lead compounds that might actually benefit the development of a structure like muscle. We can then test these, go on to test them in mice, potentially in larger animal models if they're available, and hopefully end up with human clinical trials of something that might be therapeutic. So actin Nemaline myopathy is one of the conditions that my lab has studied quite a bit over the years. This is the same diagram you've seen previously. As I mentioned earlier, we now know of over 250 different actin mutations, all of which alter the protein in a slightly different way. And most of them cause a dominant disease. So having one abnormal copy leads to enough abnormal gray balls here to cause the condition. As I mentioned, these numbers, an underestimate is roughly at 30% of individuals with Nemaline myopathy have some of these mutations. And we're now starting to understand the basis for their muscle weakness. In fact, research that's been supported by a foundation building strength done by Henk Granzier in University of Arizona and Koen Ottenheim, who's now got his own research lab in Amsterdam in the Netherlands, has helped us, and I should say that that's been research done with some of the muscle biopsies from individuals who have enrolled in our research studies, have now allowed us to start to understand how the weakness is caused. And for example, we now know that patients with mutations in the nebulin gene have thin filaments that are too short. and that leads to less overlap between the thin and the thick filaments, and therefore less generation of force. And we also now know that, for example, some of the actin mutations alter the shape of that protein and actually cause this thin filament to fall apart. So we get broken thin filaments in that case, and that's the basis for weakness in that condition. So because this is a common cause of Nemaline myopathy, we have decided to generate a series of zebrafish lines with a variety of different mutations. And shown here is the identity of 6 different mutations that we're working with. We chose these for a variety of reasons. We chose some particular mutations. This code here, H42Y, refers to the particular nature of the mutation. So what that means is a histidine at position number 42 in the protein is altered and mutated to a tyrosine. Or for example, M134V would be a methionine at position 134 changed to a valine. And just that one simple change is enough to alter the structure of the protein and cause it to not function properly. But as we see on the right here, the severity can vary. So individuals with H42Y tend to be quite severe, very weak. Individuals with M134V have a milder condition, often walk for most of their lives. Another reason we chose some of these is that, for example, there's a mouse model now for H42Y and another one for M134V. So using the zebrafish allows us to screen for thousands and thousands of drugs. Anything we find can then be tested for in mice much more rapidly and much more cheaply. So our initial experiments involved just adding in some of the abnormal protein. And in fact, we added in by microinjecting messenger RNA, which leads to the production of the protein. And we had tested 3 different mutations shown here. M134V is missing, V165M, and H42Y. The messenger RNA is injected into a fertilized egg, shown on the left here. And then we waited and we counted at what time, how soon these fish hatched. So shown at the top here, at about 4 days of life is a fish that has hatched. And at the bottom here is a fish that was so weak that it got stuck inside its egg and never wriggled enough to hatch. And you can see that in the purple line is the rate at which normal wild-type fish hatch if we do nothing to them or if we inject them with some control messenger RNA. And then these green lines and the orange line show the rate of hatching for fish that had been injected with the abnormal actin. So what we found is that when fish have a mutation in the actin gene, they have a delay in the timing of hatching, and we know that this delay is reflecting intrinsic muscle weakness. We can look at these fish other ways. This is what the fish looked like after they did hatch, or in some cases we actually could help them hatch by poking the egg with a needle and freeing the fish. And you can see H242Y, we have a very curved fish. The V165M fish is somewhat curved. It's very difficult to see on the right, but using a microscopic method, a microscopy method called birefringence, we're able to see the structure and the organization of the muscle. The normal fish has a bright staining pattern shown at the top, and each of these mutants had reduced levels of birefringence, indicating that their muscle was a little bit less organized. And then finally, we could actually count the numbers of fish that either were very severe, were mildly, moderately severe, or normal. And a remarkable thing that we found is that the level of severity actually correlated with the severity of the mutation in humans. So the H42Y fish had the greatest percentage of severe or mildly affected embryos, and the M134V had the lowest percentage of very severe looking fish. And this actually correlates roughly with the severity in the human phenotype. So this was pretty nice to see because it gave us some assurance that in the fish, these same actin genetic mutations operated in the same way that they do in humans. Finally, we made some transgenic fish. So what I showed you was injecting the messenger RNA. That causes a temporary problem, and you can see that it leads to a severe defect in the fish that we see very early on in life. But by injecting the gene instead of the messenger RNA, we can actually make a fish that will inherit that mutation and pass it down to its offspring. And I'll just go quickly through here. This shows the shape of various fish over time, and at the top is a DNA tracing that shows that these fish actually carried one copy of the normal gene and one copy of the mutant gene, because we have two of these peaks right on top of each other at the same location in the gene. Finally, we're now using a method called CRISPR Cas, which has had a major impact on the effects of our ability to target mutations. So CRISPR Cas9 is a system that allows us to use proteins and a DNA sequence that allows us to cut the DNA at a particular location. So we can actually target and find the actin gene and alter the actin gene in that particular fish at a high frequency. And there are ways to do this you can use to create mutations and potentially also to repair mutations. And I know somebody asked about work that Nigel Lang is doing. I think Stacy Cossette had sent an email out about a year ago. And this is one approach that they're considering to try and actually go in and target the Nemaline gene with normal sequences, and using the CRISPR-Cas system make a correction at the particular location where the mutation is found. So why are we doing this in the fish in particular? Well, I've been mentioning the ability to screen drugs, and this is really a very nice system when you have something like Nemaline myopathy, where the defect is in the skeletal muscle, and we actually don't see much of an effect of cells grown in tissue culture. So you need a whole organism. You can't really screen, you certainly can't screen drugs in human patients. Sometimes we'll have dog or cat models of disease, but you can't screen drugs in those either. Even in mice, it's very expensive and time-consuming to screen because you can only test one or two drugs at a time. However, as I mentioned, if we mate two fish, we'll get about 100 embryos that are fertilized. We can array those embryos into a multi-well dish here, and then just simply using a robot, add various chemicals one by one into each of these different wells of the drug, incubate, and then watch and observe the phenotype of the fish after it hatches several days later. Because the fish is clear and permeable, just simply placing the drug in the water in many cases leads to the drug getting in and potentially having an effect on the development of those fish. So an example here, we've already been successful, is with a fish with a related mutation in a gene called RYR1B, the ranitidine receptor. This causes a related form of congenital myopathy, and in fact occasionally does lead to Nemaline rods. On the left here is a video of what a normal fish looks like when you poke it and it swims away. You'll see it swims extremely fast. It's this is the normal escape response that fish use to sorry, turning my phone off, to avoid capture. This RYR1B fish, on the other hand, you can see it really doesn't go very far. In fact, the investigators that discovered it gave it the name relatively relaxed because it doesn't swim too far. It seems relatively chill. So we've done a drug screen on it, and long story short, here's the results for that. At the top two wells here, this is a photograph now of a well that's been optically tracked under a computer, and the red lines show the path of the fish over a 10-minute period as it swims around. And these are fish that were 20 days old. And you can see a wild-type control fish swims quite well, and a wild-type fish that's been treated with our drug, NIF, actually also swims pretty well. And you can see that in the two bars on the right here. Distance traveled, they've both gone about 5 1,000 millimeters over the time of that particular experiment. In contrast, our RYR1B fish treated with DMSO, this is our control, doesn't swim very far at all, and it doesn't go very far, and you can see that in the bar graph on the right. And yet when we treat it with our drug NIF, that restores its ability to swim a much longer distance, as you can see by the bar graph on the right. Some other ways of looking at this, the wild type fish tend to be fairly straight when you look at them from the side or from the top. On the other hand, the mutant fish have this sharp curve due to muscle weakness in the trunk, and you can see that when these fish have been treated with our drug, they tend to be much straighter. And again, you can see here are on the right, the control, or the RYR fish treated with the control drug, and then treated with a real drug, and you can see the improvement. So last slide before we take some questions. We're now actually Donna Gupta, who, as I said, now has her own lab, has been working with Nemaline fish. You can see that this is a very nice model. These are fish with a mutation in the Nemaline gene, very similar to what we see in humans. On the right, you can see a control fish glows quite nicely with birefringence when we put it under polarizing light. And then below, the Nemaline-related fish has very, very little birefringence, indicating that the muscle structure is highly disorganized. She's able to prove that this is actually a mutant fish by sequencing it, shown on the right here, because these fish carry a mutation changing the C to a T at a particular position in the Nemaline gene. And you can see that these fish on the activity monitor on the lower left don't swim nearly as far as a control fish. So getting back to Vandena's discovery about the Kelch proteins, about KLHL41 that I mentioned earlier, and its ability to stabilize and affect the turnover of Nemaline, Vanda has now realized that turnover of Nemaline is probably an important contributor to Nemaline myopathy, and is testing a series of drugs that affect protein stability and degradation, and starting to have some very encouraging results, discovering drugs that may actually impact the stability of Nemaline. So I'm going to end there by just, whoops, went too far. I want to show my acknowledgments and then answer some questions. And I want to just thank everybody who's been involved. First of all of you guys, the entire Nemaline myopathy community. Without this work, we really couldn't do any of this. Collaborators, as I mentioned, we've worked over the years, many people not listed here, but in particular Nigel Lang and Kristen Novak's group in Australia, Karina Walgren-Pedersen's lab in Helsinki, Kathy North, Mike Lawler, who's trained in my lab now, is at Medical College of Wisconsin, and then a number of funding agencies, in particular Foundation Building Strength. I have to say that AFBS has really been very helpful in ensuring that there's a focus on developing treatments for Nemaline myopathy, on bringing together investigators and families, and on funding some of the work that otherwise wouldn't get done. So with that, we'll take some questions, and I actually see one that has been texted to me asking whether there's any update on whether the inclusions of Nemaline rods is the cause of NM or an outcome of NM. And that's a great question, something that wasn't clear for a long time. But I think now our understanding is that Nemaline rods are probably a consequence of the mutation and not necessarily the cause of weakness. So what we can find in work that's we've done in collaboration with Kuhn Ottenheim, you can take a muscle fiber that has relatively few Nemaline rods. Kuhn is able to measure the strength of contraction of that muscle fiber, and in fact it can be really quite profoundly weak. So our thought is that the rods probably are a secondary consequence, and they're a marker of whatever it is that is leading to the weakness, but the real causes of weakness are something else. And in fact, let me just go quickly to a slide I showed at the beginning. And I'll just show you all a link to a URL. We have an animation that we created a few years ago now, shown at the bottom, www.childrenshospital.org/begs/Nemaline. And this animation actually illustrates the mechanisms that we think are leading to weakness in 3 of the forms of Nemaline myopathy, the nebulin-related form, the actin-related form, and tropomyosin-related form. And you'll see that really the weakness is a consequence of defects in the structure of the sarcomere, particularly the defective thin filament and how that impacts its interaction with myosin, which is responsible for generating the force of contraction. So hope that answers that question. Happy to discuss it more offline. I see also another question here. How long do we think it'll be before treatment starts? And that's the $64,000 question, and obviously key for everybody here. I really hate to answer these kinds of questions or give predictions, and I'll just tell a little anecdote related to that. And that is that when I started my training, I trained in the laboratory of Lou Kunkel, who had just discovered the gene for Duchenne muscular dystrophy. And this will date me, but that was in 1988. The gene was actually discovered in 1986. And so in 88, when I started, the Muscular Dystrophy Association and the press and everybody was touting this accomplishment. The gene had just been discovered and everybody was saying, well, in 5 years now that we know the gene, we're going to have a treatment. And it's now 2017, and treatments are just starting to become available for Duchenne dystrophy. Remarkable progress has been made in just the past few years. And I think that things have accelerated tremendously. I would love to say that for Nemaline myopathy, maybe within 5 years we'll have a treatment. But I actually won't say that without the qualification that it's really impossible to predict. We do have clinical trials, as I mentioned, starting for gene therapy of myotubular myopathy. And I've been working my whole career on this group of congenital myopathies with the goal of developing treatments. And so this is a very exciting time. The work we've been describing with the drugs, our hope is that the transition to clinical use might be faster than normal. And that's because we're taking an approach called repurposing. So rather than testing random chemical compounds, many of which might be toxic or not suitable to use in humans, we're testing libraries of drugs that have already been approved for other uses by the FDA. These so-called repurposing libraries then allow you, if you identify something that's beneficial, even if we don't completely understand how it's working, we do know that it's been shown to be safe and effective for another condition, and that allows for more rapid progression to the clinic. So our hope is that within the foreseeable future. Okay, so I see a couple more questions here. Understanding that NEB is a big gene, but given that many of the population has very small NEB defects, do we think researchers are rethinking treatments like CRISPRs for NEB treatment? Well, so the power of CRISPR is the ability to go in and find that 1 or 2 base pair change. And previously, if we wanted to change a particular base in the genome, we were able to do it in cells in a dish, but it was a very rare event. And you have to have some selection where you basically change the DNA in hundreds of thousands of cells and then find some way to kill off all the ones that don't have the change you want and let the 1 or 2 grow out that you were looking for. With CRISPR, we can do this much, much more efficiently. So now we can affect the change in maybe 1 out of 100 cells. The problem is that when we transition to the whole body, it's very difficult to get the efficiency high enough to have an impact. And there's been a lot of excitement and work developing treatments or people trying to do this in mice with models of Duchenne dystrophy. But frankly, it's been working only at a very low efficiency. So it isn't so much the size of the NEB gene that's the problem, That does make it difficult to find the mutations in the first place, but once we know what they are, CRISPR is able to target just that small change. However, the efficiency is still not what we would like it to be. So then some more questions here. So it's a question from somebody who has one of the Nemaline mutations identified and the other one not. And this is a big issue that we're trying to deal with. So because of the size and complexity of the Nemaline gene, it is there are a lot of changes that are not findable even with the current whole exome sequencing approach. So we have been trying to identify, use whole exome sequencing to identify as many mutations as possible. And in about 2/3 of our patients with a Nemaline mutation, we can find two, one from mom, one from dad. But we still see that about 1/3 of those patients, we can only find one mutation, and yet we know there must be a second one lurking there somewhere. One of the problems is that Nemaline is complicated and has a repeating structure. So there's a region in Nemaline where the same exact sequence is repeated 3 times in a row. The problem is that that makes it almost impossible to locate a change when we find it. We don't know which of these 3 regions it is. And the signal from the 2 unmutated regions actually sometimes swamps out the signal from the 1 mutated region. The other issue is that because the gene's very large, there are other types of mutations, larger rearrangements, or even deletions or duplications, and the methods we use for next generation sequencing today just don't identify those types of changes. So we and others are working on more efficient approaches for looking at these large genomic changes. So hopefully we'll get there. There's a lot of effort in that area, but it's still a problem. And if anybody wants specific updates, reach out to me offline, and we can check on the status of a test either in my lab or in Karina's lab in Helsinki. Question, why doesn't NM affect non-skeletal muscle fibers? What we understand now is that all these genes, and I didn't emphasize this, but each of these genes encodes a protein that's used only in skeletal muscle. And so that's the reason. So for example, there's no Nemaline in the liver, there's no Nemaline in the brain, or in other parts of the body, only in skeletal muscle. That's not true for all muscle conditions, but in particular for Nemaline myopathy, that's what we found. The Kelch proteins might be slightly different. Some of those are expressed in other places, and some of the patients with the Kelch mutations actually have very severe disease that may be affecting others. So I'm not going to say 100% that all cases are exclusively skeletal muscle. But by and large, the thin filament, which I showed you, that's a structure unique to skeletal muscle. And that explains why we don't have problems with other organs. Question about registration and enrollment. So this could be confusing to folks because there are all these different research initiatives and registries and so on. My answer is always enroll in as many different initiatives as you feel comfortable, but also understand that there's a very good chance that as long as you stay engaged, being involved in any one will help give you the benefit of hearing about advancements as they come. The CureCMD has a registry called the CMDIR, the Congenital Muscle Disease International Registry. That is a contact registry and is combined with a tissue bank and repository run by Mike Lawler at the University of Wisconsin, Medical College of Wisconsin in Milwaukee. And Mike, as I mentioned, actually trained in my lab. He's now been out there for, I think, more than 5 years. We all talk to each other. We don't share patient identifiers, so if you sign a informed consent form, for example, for my study, it actually allows us to share information, but not identifiers. So I'll never say Judy Jones is in my study to Mike unless whoever that is has given me permission to do so first. But we will also all coordinate. And so as new initiatives become available, we let everybody know. AFBS is a great central clearinghouse for this. And in fact, AFBS, as you may know, has supported the Nemaline Myopathy registry in Mike Waller's lab, and tissue bank there, which is also connected with the CMDIR tissue bank. So, sorry, I'm kind of rambling here, but I think essentially register in each of the places where you feel comfortable, but don't feel like you have to do everything if it's too much. Sarah, have you been looking at these? Are there any questions I missed?
Yeah, this is Sarah. So I just know that we're running a bit long. So what I could say is that if people have questions that they weren't answered, that they could go ahead and contact me, and I can get together a list of questions if they still have remaining questions after the webinar. And or also, I know Casey put her email address in the presentation, so you can reach out to Casey. And so just wanted to, again, thank you. Would you mind going back just to wrap up and put back up a bit about the Nemaline Myopathy family gathering registration? And then if people are able to make that, we have this meeting getting together in New York City. And if you're able to come, that would be fantastic. And you can, we can talk in person about some of these issues and get everybody up to date with contact information and maybe answer some questions and talk about it together as families, and just also have just a fun time together in a social event. So the registration is there. And I want to thank you, Casey and Alan, for taking time out of your Saturday to be with us. And we'll make these available, this webinar available online for people, so that if they want to go back and review parts of it, or if they might have additional questions, that they can reach out to us with questions related to today's topic. So I'm going to go ahead and stop the recording and just thank you so much.